Okay, great. Well, good afternoon and welcome to everyone. I see some familiar names and faces. We're so delighted that you could join us this Friday afternoon. As many of you know, at the Melanoma Research Alliance, advancing research is at the heart of what we do. In fact, we recently announced our 2025-2026 request for proposals with plans to support at least $11.5 million in new funding for innovative melanoma research projects. Today, we're thrilled to focus on one of the most exciting areas of science driving progress, and that's the LAG-3 and how combining it with PD-1 is helping more patients respond to immunotherapy. So we've got some really exciting things to chat about. My name is Dana Dayton, and I'm the Director of Communications and Engagement at the Melanoma Research Alliance, and I'll be moderating today's discussion. We'd like to thank you for joining this webinar, Unlocking LAG-3, New Frontiers in Combination Immunotherapy for Melanoma, another in a series of patient-focused webinars designed to educate, engage, and empower the melanoma community. We'd also like to thank Regeneron for sponsoring this installment of our MRA Meetup series. You can visit curemelanoma.org/blog and click the Patient Webinars tab to view a library of our past events. Before we get started, we'd like to review a few housekeeping items. By joining this webinar, you understand that this is a public event and is being recorded as a resource that will live on MRA's website and may be shared on social media. This session is for educational purposes only and should not replace professional medical advice. Please consult your healthcare provider about any questions specific to your care. It is your choice to be on or off camera and how you would like your name displayed. Please keep your line muted and silence other devices. If you would like to ask a question live, please use the raised hand feature in Zoom so that we can call on you. So I think that's about it. And now I am going to get started. It is my incredible pleasure to welcome today's expert, Dr. Hussein Tawbi, who is a professor and executive director of the Immuno-Oncology Clinical Drug Development Program at MD Anderson Cancer Center. He also co-directs the Andrew M. McDougall Brain Metastasis Clinic and Program. Dr. Tawbi is a senior clinical investigator and global leader in melanoma clinical trials with a focus on immune checkpoint blockade and novel combination strategies. He is passionate about improving access to clinical trials and expanding treatment options for patients at every stage of their journey. Dr. Tawbi, thank you for being with us today. We're excited to dive in, and it is such an honor to have you.
Thank you so much, Dana, and thank you for the very generous introduction. I really appreciate the Melanoma Research Alliance putting this together, and I know you and Renee have worked very hard to kind of prepare the content for this. I want to welcome everyone, and should have probably called it Happy Hour with Tawbi on Fridays because it's pretty late, but really great to see some familiar names and faces that, again, really great to see everybody being here. So I'm going to give you a little bit of a background about why I do what I do. And I think, well, the first thing is for most of you that know me, I'm just a big nerd and I just love science and learning things since I was a wee bit, kind of kid. And so as I got into oncology and realized that it's a place where you really can form a team with your patient to try to beat a common enemy, it became very clear to me that we just didn't have enough things to help cancer patients. And I started working in melanoma at the University of Pittsburgh because I was just very impressed by the research that was being done in melanoma. But I tell you, that was in 2005. And it was days when things didn't look that great for melanoma. And within a very quick few months, I became a phase 1 doctor because our first line therapy at the time was clinical trials, and more specifically phase 1 and first in human trials, because we were trying to find something that could change the course of melanoma natural history. And it really took a few more years before we went from having really not great prognosis to starting to cure patients with checkpoint inhibitors. So when Dana, you say I'm passionate about this, honestly, just observing how you can go from a discussion where things may not end well to say, look, we think we have a pretty good chance at curing you. That is a dramatic moment in cancer history in general. And I'm really proud of the fact that this happened in melanoma first, and it continues to be the place where we learn how to do things and then can translate them to other cancers. And so over the course of the last 15 to 17 years now, it's been my absolute honor to participate in some of these trials and lead some of the developments in that space. But you also mentioned something very important to me, which is the fact that as we started getting these cures and ability to really help patients long term, I started getting really unhappy when I saw patients being excluded from trials or not treated because the data was not generated in those populations. And that included patients with autoimmune disorders and patients with brain metastases and so on and so forth. And so it became a bit of a mission to try to focus on these populations that are potentially higher risk, but in which checkpoint inhibitors can still make an impact. And so yeah, I mean, I think that's kind of very briefly how it ended up being that LAG-3 in combination with PD-1 was one of the newer checkpoint inhibitors that we got involved in early on and kind of led its development all the way to phase 3. And I'm happy to discuss it today and educate as much about it and share with folks what the future may hold for this for the LAG-3 based combinations.
Wow, yeah, that's an amazing setup. Thank you for walking us through all of that. Indeed, we are so lucky to have all of the advancements that we've seen over the last 15 years or so. But as all of us know, there's so much more that needs to be done because there are patients that are not benefiting from the current treatments available. So everything that happens, as they say, happens for a reason, but it all starts with clinical trials. And just to set the stage, we would love to sort of level set and talk a little bit about why clinical trials are so important to moving the field forward. And maybe you can walk us through a little bit, sort of the basics, like what does it mean to be in a head-to-head trial or have different arms and things like that. There are always questions surrounding that. And even though I know we have a really educated audience, it never hurts to review some of that.
Yeah, no, absolutely. I mean, and it's kind of interesting when I was just saying 15 years ago, the first line option for a patient with melanoma was a phase 1 clinical trial because you look at what are the standard of care options that are available today, and even when we have really great outcomes, we still have about half of our patients that don't do well, and our current approved combinations include ipilimumab, nivolumab. I always call it just ipi-nivo, nivolumab and relatlimab. Then you have TIL therapy. And for those patients that have a BRAF mutation, which is about half of all patients, BRAF and MEK inhibitors. And that's it. I still have one finger on my one hand. So that means we don't have enough options. We don't have enough shots on goal. And so the ability to try to, kind of improve the outcomes of those, 40 to 50% of patients that are currently not responding can only come from treatments that are not standard. So by definition, access to treatments that are not standard means that they're investigational. And so the only way to be able to access them is to participate in clinical trials. Now, having said that, we don't run a clinical trial with a drug that we don't think has promise. We actually bring it forth because the science behind it seems that it may be capable of doing something we're currently not able to do. So by definition, it's innovative. By definition, it is promising, which is why we're willing to put it in patients. And again, by extension, it could be something that could give us more shots on target and can, reactivate the immune system and allow for long-term control. And I say this today, so clinical trials is where we have all of our innovative and promising therapies. But if you think about it, the current standard that I was just mentioning, all those four things on the one hand, they started off being clinical trials too. So when I was in 2005 a fellow and then on faculty in 2007, those did not exist. And it is patients that participated in clinical trials early on that, helped them become the standard of care. So the way I think about it is today's clinical trial is tomorrow's standard of care. And some of you may have heard recently about, Jerry Jones's story, for instance. And I think it's a great one. It's exactly, he chose to talk about it 10 years later when he participated in the early trials of single agent PD-1. It was a clinical trial. It was not a standard of care. And it led to a cure. And here we are 10 years later learning about it. So it is exactly kind of illustrates that exact point, that what is today's clinical trial could become what everybody else gets treated with in the future. And so I think it's exciting to be part of trials. I think it is also a way to get the best possible outcome for yourself, and it's a way to help the entire field advance.
Thank you. Yeah, that's really important. For people listening, how can they best access clinical trials? Do they need to be at an academic center, or might their community physicians be able to help make that trial accessible if they're interested in one?
Yeah, so that's a really great question. I mean, naturally the kind of there's a more density of trials at an academic center, meaning there's going to be probably a larger variety of trial at an academic center because, we participate in phase 1, phase 2, and phase 3 trials. We run neoadjuvant trials, all that kind of stuff. But there are types of trials, especially usually ones that are later stage, like phase 3 trials that maybe may have community sites as accruing sites, and so patients may actually be able to get access to them, at a community site. So I think the important part for me is to ask the question. Whoever you're seeing, just ask the question: do you think a clinical trial makes sense for me, or do you have clinical trials that make sense to me? And when I discuss clinical trials with my patients, I share with them what is the standard available. And I also share with them why, if I consider a clinical trial for them, it's because I think the clinical trial has the potential of being better than the standard. If I'm putting myself in that one situation where the standard may be better, and I wouldn't propose the clinical trial. And so that's kind of where I would recommend people think, ask the question, and just be open-minded about, kind of seeking second opinions. I mean, the entire kind of academic community is always prepared to hear other folks' ideas and opinions, and that should, being at a community or an academic site should not stop you from considering to ask somebody else. You also did ask me about the head-to-head studies, and maybe we should spend a minute talking about what kind of trial are we talking about. So when I talk to patients about trials, commonly I hear things like, is there placebo? Is there blinding? Am I going to not get the drug? And I think those are important questions. The vast majority of trials are not blinded, and the vast majority do not have placebo. So, but there are trials that are randomized and blinded, and if there's a component that's placebo, it's usually not the only thing you're getting. So, even Relativity 047 that we'll discuss later today that led to the approval of nivolumab and relatlimab, it was randomized, blinded, but patients on one arm got nivolumab and relatlimab, and the other arm got nivolumab, which was the standard of care at the time. So even randomized blinded trials don't mean that you're not going to get the right treatment. Now, there are some trials in which we're comparing an entirely novel combination to the current standard, which would be, basically the one I just mentioned, nivo-rela versus nivo is a great example. And there are situations in which we're comparing drugs that target the same pathways, but we're trying to see which one of them is better at targeting that pathway. So those are usually called head-to-head trials, meaning that an example for that was the Columbus trial, which compared encorafenib and binimetinib to encorafenib alone and to vemurafenib. And so the arm where patients were getting the BRAF inhibitor encorafenib versus the BRAF inhibitor vemurafenib, they both target the same pathway, but it was comparing head-to-head a BRAF inhibitor to a BRAF inhibitor. Similarly, we have a clinical trial right now that's running comparing nivolumab and relatlimab, which is a LAG-3 PD-1 combination, to fianlimab and cemiplimab, which is a PD-1 LAG-3 combination. So both combinations target the same pathways, but there are reasons to believe that one of them may be better than the other. So we just ask the question in a randomized way. We take patients and we say, we'll flip a coin and you'll either get this standard or this very similar combination that could be slightly better than the standard. So I hope that addresses the question, Dan.
Yeah, that was really great. Thank you. I think I even learned a little bit, and I've been around the block a few times with this. What I love about clinical trials is that, you can isolate a variable and get a very clear answer, once the data is collected. And I think sometimes, people are a little curious about, why do I have so many check-ins and so many lab draws and so many imaging, dates and things like that. But I think, without all of that data, it wouldn't move along, as precisely as we all need it to get to the right answer. So that was really great. Moving on, I want to jump into, the LAG-3 and the science behind the trials, which is why we are here. I absolutely love the fact that Regeneron has these 4 trials that are active right now. So I'm hoping that we can talk about some of them. I know most of the community is familiar with PD-1 and CTLA-4, but maybe less familiar with LAG-3. Can you set the stage here and tell us a little bit about LAG-3 and why it is being studied?
Yeah, I mean, if you're familiar with PD-1 and CTLA-4, then LAG-3 is not very far. So, I mean, the idea starts from why do we target the immune system in the first place? I mean, if you think about how cancer develops, your immune system is surveilling your body every day, and it literally almost touches every cell in your body every day and checks if it's self or not self, and whether it should keep it or destroy it. And so even developing melanoma in the first place and it going all the way to become a detectable malignancy, I tell my patients it is almost a disease of the immune system, because the immune system was checking that one cell that was already thinking about becoming malignant and somehow decided to turn the blind eye and not destroy it. And that's why it grew to become a full-blown cancer. So trying to make the immune system correct that mistake and make the immune system see that tumor is at the heart of all of immunotherapy and specifically checkpoint blockade. So we realized over time that there are several checkpoints that prevent the immune system from doing its job. So CTLA-4 prevents the immune system from recognizing a variety of antigens. And so when we give CTLA-4, it allows the immune system to see more antigens and get more activated against them. And PD-1 is one of those things where the immune system has already recognized an antigen. The T cell is already antigen specific and is prepared to go and kill the cancer cell. But in the tumor microenvironment, and the cancer cell is able to block the T cell from doing that using the PD-1 receptor. So when we give a PD-1 antibody, we allow this antigen-specific T cell that already knows the tumor, we allowed it to be more efficient at killing it. Now, having said that, sometimes T cells recognize the tumor, they're attempting to kill it, partly because of PD-1, they may not be able to. And then if the tumor persists and is there, and the T cell is continuing to try and try and try, at some point, the T cells recognize that they're not going to be able to succeed, and they get to this state called T cell exhaustion, where they couldn't clear the antigen and they stopped trying as hard. And so that T cell exhaustion phenotype is something that we recognize through a T cell that has PD-1 on it. But as we learn more and more about the immune system, we also recognize that an even more exhausted T cell has one more checkpoint on top of it, and that checkpoint is LAG-3. So if you have a T cell that has both LAG-3 and PD-1, it's going to be a lot more exhausted than a T cell that has PD-1 alone. And that is why targeting LAG-3 with an antibody, like we target PD-1 with an antibody, made a lot of sense. Now, the trick with LAG-3 inhibition was actually that if you try to block LAG-3 on its own, it's kind of, it doesn't do the trick. You have to actually block both LAG-3 and PD-1 at the same time. And by doing that, that's when we unlock this additional potential for T cells to work. And so now that's why the two drugs, or kind of that combination, is approved only as a combination. Like, you can't go and get relatlimab, the anti-LAG-3 antibody, on its own, because it's not going to be effective. But if you put it with PD-1, then you get this additional efficacy. And because it's these specific T cells, we're very excited that this additional efficacy doesn't come at the cost of a lot of more toxicity, which is something that we haven't been used to. But I'll address that in a minute. But I just will stop here just to make sure that this is clear so far.
Yeah, that's very clear. Thank you very much. I feel like it's in my mind, I start to think of like a soccer team or a football team, and like you're bringing your best players on the field to do like the job that they're supposed to do uninhibited. So, thanks for talking us through that. So now we know, I think you mentioned this a little bit, like nivolumab plus relatlimab, which is Opdualag, was the first LAG-3 PD-1 combination to be approved. How does Regeneron's fianlimab and cemiplimab program build on that?
Yeah, so again, to further kind of describe the nivolumab relatlimab combination, or Opdualag, it was really great to see that it's so much more effective than single agent PD-1, and that was how the trial was designed and randomized, to compare it to single agent PD-1. So we know that we improved the response rates from 30 to 44%. We know that we improved the progression-free survival almost by 25%. We doubled the median progression-free survival. And there seems to be an impact on overall survival as we follow this study for a longer period of time. Now, the interesting part is we had lived for 10 years with ipilimumab and nivolumab, and we had lived with the fact that the only way you can get more efficacy over single agent PD-1 was the combination of ipi-nivo, which is great. We love it. We still use it every day. But we also knew every time we reached out to it, we're going to have to contend with toxicity that goes from 15% with single agent PD-1 to 55% with ipilimumab and nivolumab. So that really limited our ability to do things. It made us have to use steroids in patients where, as you all know, we don't like steroids because they're literally the antidote to the immune system. So it's always been very challenging. But when we see what happened with nivolumab and relatlimab, we were really excited because we unlocked this additional benefit. We got all of this increased, activity, and the toxicity rate went from 15% to 22%. So it wasn't a massive increase. And so we're kind of excited that now we can offer it to more patients, especially if there are issues with any other or kind of performance status issues or tumor burden issues. And we also know that we don't have to use as much steroids when patients develop toxicities, which to me is kind of the more exciting part. Now, again, when you have a combination that works and targets a specific pathway, I think relatlimab and Opdualag really have just validated that that's a good therapeutic target. That's the most important aspect that it did. So trying to target it with other antibodies makes a lot of sense. So cemiplimab similarly is a PD-1 antibody that seems to work similarly to single agent nivolumab. So fianlimab was the new LAG-3 inhibitor, and the early data from about 100 patients, 98 to be exact, from fianlimab and cemiplimab looked like it actually is breaking a higher ceiling for response rates and for progression-free survival. So we're seeing response rates closer to 60% and median progression-free survival that's up to 24 months compared to 11 months with Opdualag. So the single arm data looks like it could potentially be more effective, but we don't know that, and that's the thing. That is why we need to be able to generate randomized data, and especially if we can generate it in a head-to-head way. So you mentioned the 4 trials that Regeneron is doing. So I think, the first trial is trying to get fianlimab and cemiplimab potentially FDA approved by comparing it to single agent PD-1, very similar to what a Relativity 047 did in the first line metastatic setting. So if that trial, ends up being positive, then we may have that combination FDA approved. Another trial is using the same combination in the adjuvant setting. There's a trial using the combination in the neoadjuvant setting, which you may know this is for patients with bulky nodal disease. It's before they go to surgery, which we've actually done a trial at MD Anderson with nivolumab and relatlimab and showed that it's actually a highly effective combination in that setting. So that's the third trial. And the fourth trial is the one I already mentioned, which is the head-to-head comparison. And I'm excited about that one because we're going to be able to know for a fact, is fianlimab cemiplimab truly a better combination for LAG-3 inhibition than nivolumab relatlimab? And I'm excited about this trial because very few times in a clinical trial do you feel like whatever the answer is going to be good for patients. If it's going to be that, yes, the new combination is better, then exactly that. We're going to have a new combination that's better for patients. And if it shows that they're about the same, then now we have 2 combinations to choose from. So either way, I think it's going to be a good outcome.
Yeah, that's great. And I love how not only are you looking at different combinations, but you're looking at different settings so that more people can potentially benefit. So would you mind running through exactly which of the 4 trials are still enrolling patients and if any of them are open to some of our rare melanoma patients like acral and mucosal or uveal patients?
So I know the neoadjuvant trial is ongoing, and so when we say a trial is ongoing, let me just be clear on one fact. So sometimes a trial could be ongoing, but the accrual has finished. So the first line metastatic trial, the accrual is already finished, but the trial is ongoing because we're looking at the outcomes of those patients, right? The neoadjuvant study is accruing right now, and the head-to-head trial is accruing right now. And we do have, so nivolumab and relatlimab had about 18.9% of the patients that were on the phase 3 trial had acral or mucosal melanoma. So it was a pretty large representation for those patients. And the response rates were in the 30% range.
Wow, that's fantastic. Thank you for sharing that. So let's talk a little bit about, which patients should be asking about which studies. Many of these trials are focused on patients at high risk for recurrence, or those that have not responded well to previous therapies. Can you give us a few, top line tips on what patients might want to ask of their physician to see if they're right?
Yeah, so one of the interesting things with LAG-3 inhibition was what we learned from the phase 1 trial with Opdualag was that you want, because of the mechanism and what it targets, it may be more effective in earlier stages of the disease. So if patients have already received prior PD-1 or prior ipi-nivo, the response rate is about 12%, which is not really dramatic. It's still useful, and I still use it regularly, but it's not as, kind of impressive. But then if you go to the first line metastatic melanoma, so previously untreated, metastatic disease just, kind of confirmed, it seems that that setting has better activity than the patients that are already progressed on PD-1. And by the same token, we actually asked the question, can we use it in the neoadjuvant setting where the tumor is still localized, surgically resectable, but has not been resected yet? And that is a situation where it's also less, the immune system is less suppressed. So when we used Opdualag in about 30 patients in that setting, the response rate went from 44% in the metastatic setting to 63%. So there seems to be a little bit of value to earlier lines of therapy. We also feel like it's very important to have the tumor in place when you treat with PD-1 and LAG-3 combination. So folks may have heard that the adjuvant trial from nivolumab and relatlimab compared to nivolumab alone did not hit its primary endpoint, and it doesn't seem that treating with the combination after the tumor has been resected may not be as impactful. So we really feel the presence of tumor is going to be an important determinant of having the tumor response.
So these 4 trials really have the potential to be pretty foundational for what's next. And I'm sure as a scientist, you're already thinking ahead. What would you like to try next? Any potential things that you see on the horizon that would be the next step?
Yeah, well, I can tell you that we don't kind of sleep on our laurels, so to speak, and we're just very happy that we have a new combination that is more effective and potentially safe for our patients. But a couple of things. First of all, I would say that we still have populations in which there has not been proven benefit because they have not been included in clinical trials. So for instance, one very important population is the melanoma brain metastases, for instance, where we know that, ipilimumab and nivolumab was tried in a phase 3 trial. Patients weren't included, so we ran a separate trial with ipi-nivo for brain metastases where we showed excellent activity, and now that became the standard of care. Similarly, with Relativity-047, while we had benefit, we did not actually allow patients with brain metastasis were not allowed on the trial. So we're trying to generate data in that space to show that that combination may be effective in the brain as well. So there's a trial that's running at MD Anderson right now looking exactly at that population. We're definitely interested in acral and mucosal melanoma, as you said. So the trial did include some of those patients, but what I'm really excited about is when you have a combination that is not very toxic, so 22% is actually really manageable toxicity, it actually allows us to layer on more drugs in the future. So the ability to combine with other agents exists. And so one of the immediate ones is obviously combining with IPI, right? Because IPI is the other drug that's around. So we have a small phase 1 trial with about 46 patients from BMS, where they showed that if you add IPI at 1 milligram per kilogram every 8 weeks, that the toxicity is not terrible, about 39%. So it increases, but not dramatically. And the efficacy from that study looks like the response rates are higher, the PFS are higher. So that is one of the most kind of the lowest hanging fruits, so to speak. I'm personally working on optimizing the dose of IPI to be given, simply because I don't like the one every 8 weeks. It feels like it's, for multiple, kind of reasons of how IPI works, that we may be better off giving it a little more frequently early on. So we're working on kind of optimizing the dose and schedule of IPI to be combined with nivolumab and relatlimab. So that's kind of a general flavor. I could go into more details, but the idea is new populations that haven't been studied and new combinations.
Great. That's really interesting. And we haven't talked much about biomarkers. Are there any unanswered questions around those that you think might enhance any of these studies?
Yeah, I mean, we've LAG-3 in its early days, we were kind of excited when we saw in the second line trial that there was some association with LAG-3 positivity, like being LAG-3 positive versus not. So we included that in the phase 1 trial, phase 3 trial. We stratified by it, thinking that we would see, kind of maybe a new biomarker, so that we might find a place where if you're LAG-3 positive, I'll give you nivolumab and relatlimab. It actually ended up not mattering as much as, that's why science is exciting, because you think something and you have to rigorously test it, and then you may find out that your original thought was not necessarily the right one. So LAG-3 positivity ended up being more prognostic. So patients that have LAG-3 may do a little better in general, but they do better with single agent PD-1 as well. So it wasn't enough for us to say we should test it to decide if we're going to treat or not. So in terms of pre-treatment biomarkers, we haven't made major progress. There's a couple of things like interferon gamma signatures that are coming through that I think may be relevant, but they're not ready for prime time. We've been learning a little more about the value of monitoring ctDNA in the blood, so that's circulating tumor DNA. And for me, that is the data from that is also emerging. It's not ready to become kind of a standard of care practice. The interesting part with that is that it's more of an on-treatment biomarker. It's not as much of a pre-treatment biomarker. So we're not in a place right now that we would check something and decide to give it to you or not give it to you based on it.
Great, thank you. Really informative. So, I know we have a lot of patients on here that are absorbing all of this information today. And as we mentioned, this website is going to be recorded and on our website, for, in perpetuity. And there are going to be other people that are starting to look at it and evaluate it, down the road. So, what would you tell any of the patients that might be listening or watching later, if they're interested in exploring these trials, where should they start?
Well, I mean, I think very importantly, just learning about things and educating yourself as much as possible. So that's what makes, the Melanoma Research Alliance and your website, I think, is going to be very helpful. I also would be I recommend being very clear and direct with your physicians and asking the questions, are you aware of trials? Are you participating in trials? Importantly, I think, again, we have really great standards of care already. I know that, we can treat patients with combinations that are highly effective. You have to discuss, the safety, obviously, of different combinations and what would make the most sense for you. The other thing I will say is whatever you read online, you really should check with your physician anyway, because there's so many nuances around your specific case that cannot be represented in anything you read on a website, right? And so there are always, history, there's always, specific conditions that you may or may not have. There's characteristics of your tumor, something about your scans. There's very specific things. And I rarely see a patient in the clinic that will be exactly the same as any other patient. Actually, I congratulate those patients, by the way. I tell some of my patients that, I know everybody wants to think they're special, but yours is a run-of-the-mill melanoma, and that's not common. So, yeah. So, I mean, I think it's really very important to recognize that, the specific clinical setting may dictate a certain path, and that sometimes the standard of care is the best thing to do up front. But
Yeah.
I really think you should ask about the availability of trials, the ones that are available, what makes them appropriate for you or not. And again, be open-minded to ask in more than one center, especially if you're at a community center, to consider, kind of learning from an academic center what's available at the time.
Yep, absolutely. My physician always says, everybody has a unique biology, so you have to remember that. Um, so thank you. That kind of wraps up our formal, questions and answers that we wanted to put out there, but we already received a couple of questions via the registration link. So if it's okay with you, Dr. Tawbi, I'll ask you some of those. So the
Absolutely.
first one is, will LAG-3 PD-1 replace Ipi-Nivo, or will there always be a place for both?
Yeah, no, I don't think it would replace it as in it becomes the only thing we would do. I think Ipi-Nivo is a very effective combination, and I use it regularly. And for me, they're not exactly interchangeable, but they're two options that I discuss with patients, and that's when I take the nuances into consideration, right? Like there are some characteristics that may make me feel like we really need to do Ipi-Nivo versus others in which I would choose nivolumab and relatlimab. So it's two treatment options that are available. They're not in competition to each other from my perspective. It's more of a what is the more appropriate thing at this particular moment in time. We're also trying to learn a little bit about what happens in the sequence. And I think, that's one thing I've been trying to really push hard for, that we need to run a clinical trial with the sequence. We know that patients that have already progressed on PD-1, and we showed some data from 047, if they progress on nivolumab and relatlimab, they still have a 20 to 25% response to ipi-nivo in the second line. The flip side is not necessarily true. So nivo-rela, as you heard in the second line, it looks to be about 12%. So I take those things into consideration, but I also, one of the tenets of oncology is you use your most effective combinations up front. So, there's a lot of reasons to consider kind of a one or two, or one or the other combinations up front. One of my goals of trials is to make sure that the decision is made based on evidence, not just based on the lack of evidence. Like in melanoma brain metastases, the information about nivo-rela just doesn't exist. So a physician cannot actually compare those two in a patient with melanoma brain metastasis. And at this point, they have to use the standard, which is ipi-nivo.
Great, thank you. So we talked a little bit about, who would be included in these trials, and here is a very relevant question to many who are looking for answers. It says, I'm really interested in how your new research could change what's possible for patients with melanoma brain metastases.
Yeah, no, I mean, that's exactly what you were hearing. We're kind of looking at newer combinations. We're thinking about ways that we could. One of the big things we deal with in melanoma is melanoma brain metastases that result in symptoms, which require patients to be on steroids. And in that case, it makes really immunotherapy at a very significant disadvantage. So some of the things we're working on is how do we manage brain edema without the use of steroids so that we can use immunotherapy as well. So, you mentioned in my introduction that I co-direct the brain metastases clinic, and I'll tell you that it's not just a clinic, but we actually have a research program around it where we're studying we're profiling brain metastases, trying to understand their kind of genetic underpinnings, so to speak. We're running clinical trials in those populations, and we have preclinical models to try to bring forth newer therapies. So it's an area of active investigation. The most important thing for me is those patients should no longer be excluded from clinical trials, and they should be involved upfront and early on. And as we do this, we learn about this population along the way, as opposed to having to do the work after, kind of after approvals.
Great, thank you. When we were speaking a little bit earlier, you mentioned some of the response you're seeing from acral melanoma patients using the anti-LAG-3 and PD-1. Can you just review that again? I want to make sure that we get to that question.
Yeah, I mean, so again, speaking of populations that are not very well represented, I mean, I think we probably need specific trials for those patients. I was very proud of the Relativity-047 trial for allowing that large of a population, almost 19%, 18.9% of the entire population was acral and mucosal. So it looked like the combination is capable of inducing a response rate of up to 30%, which is still less than cutaneous, but not surprising, unfortunately, because acral and mucosal melanomas don't have as many mutations. They're not as foreign to the immune system as cutaneous melanoma. So generally, checkpoint inhibitors alone may not be capable of kind of reversing the entire kind of resistance to immunotherapy that these tumors have. So my hope is that this safe combination will allow us, as I mentioned before, to add other drugs on top of it in the future, and potentially start helping those patients even further.
Yeah, given the difficulty in treating these cancer patients, it's still an important number that we're seeing. So that's really great. Here's another one. Does the combination show any more promise for low tumor mutation burden cancers?
Yeah, not exactly. I mean, I would tell you that when we were looking at the biomarker data, which we also published on Relativity-047, it was interesting to see that we saw a differential effects in patients where there was less CD8 positive T cell infiltration. So it did look like this combination may be able to bring more T cells into the tumor, and it works in tumors where there's not as many T cells. Usually with lower TMB, we see just checkpoint inhibitors generally work less effectively, but in that population, the combination was still better than the single agent, if that helps answer the question.
I think so. Thank you. Are all LAG-3 inhibitors the same?
That's a really great question, and we don't know. I mean, every antibody, so those are antibodies as it stands, that like fianlimab and relatlimab are two, we call them monovalent antibodies because they only target one receptor. The antibodies, when they're maybe made to different parts of the protein. We call these epitopes. So there may be a different binding. And again, slightly different affinity. So that may be a nuanced difference that may not translate clinically into a therapeutic difference, but that's why we're running this head-to-head trial, because if this antibody is actually, or fianlimab is better than relatlimab, then we'll be able to see that in a randomized clinical trial.
Right.
Now, just having said that, there are other ways to target LAG-3. We've seen some bispecific antibodies, which means it's one antibody that can target both PD-1 and LAG-3. There's been other kind of variations to the theme on bispecifics. And to date, obviously, the only one that has been approved is relatlimab, but I sure hope we can find ones that are even more effective. So that would be the goal.
Sounds like there's a lot of great potentials coming from this, so hopefully so. So I've got one more, and then if anybody has any questions, we can get to those. The last question that came in during registration, any thought to adding a CK or c-KIT inhibitor to this mix for relevant patients?
Yeah, I mean that's a good question, and it will depend on again running a trial in that population. So when we add drugs to each other, especially when we add targeted therapy on top of immunotherapy, we have to run safety trials first to make sure that we got the right dose and we don't cause undue toxicity, especially liver toxicities and other toxicities. So I think it's a reasonable question, and I am personally not running a trial like this, but I hope some of my colleagues are. And if they're not, maybe we should tell them a very smart patient came up with a great idea.
I love it. I love it. If there are no other questions, I know it's a Friday afternoon. We will go ahead and wrap it up so people can get on to their weekend plans. But this has just been absolutely fascinating, Dr. Tawbi, and we are just so grateful that you took the time to share your expertise with us. I've learned so much, and I'm sure others have, and it's going to be a really important resource for people to check back in on. It's really exciting that the field is growing so quickly. Um, so thank you again. And then thank you to all of the patients and caregivers who are taking the time to be with us now, who participate in clinical trials and make all of these advancements possible. We wouldn't be where we are without any of you today. So thank you. And I say that from, a very personal standpoint too. And then finally, we want to thank Regeneron, our amazing and very generous sponsor, for allowing us to do this. You're bringing science to people that really matters and saving lives. So we appreciate this opportunity.
Thank you, Dana. I'm just going to take a moment as well to thank you and Renee for organizing this, and the Melanoma Research Alliance for really being a leader in educating patients, raising awareness, and actually investing in science too. So thank you very much for that.